Hey, Mr. P here. In this video, we're going to talk specifically about enzymatic activity and the roles that particular enzymes play within the overall process of human digestion. One of the most important, or if not the most important aspects of enzymatic activity is the result of the pancreas. Okay, the pancreas being the single most important organ when we talk about enzymatic activity and we talk about employing enzymes to break down food during a process called digestion. A couple of the supporting structures that need to be explained before diving into enzymatic activity in more detail is obviously the role of the gallbladder as well as the role of the liver. Okay, now the liver is obviously not contained in this particular picture, but these hepatic ducts are actually ducts that are leading up to the liver. Okay, and so the liver is sitting up here. It's going to feed down through these ducts, meet with the gallbladder at this particular junction, and then will kind of dump the contents into the duodenum, or the upper end of the small intestines, through this particular duct, which is called the major duodenal papilla. The pancreas also sits in this particular location and employs its own produced enzymes through a secretion called pancreatic juice, the contents of which we'll talk about in a minute. But it's going to dump its contents into the duodenum through two ducts, the minor duodenal papilla and the major duodenal papilla. Both are ducts that are going to secrete enzymes that are entering the digestive system post-stomach, and that's important. Okay, the stomach, remember, is incredibly acidic. It has a pH of 2. We want to obviously kind of kill all of the microbes that might be present in food or out of, or on the food. And so we send all of that food material down the esophagus, kind of dump it into the stomach to store it, but also to fold in the really harsh gastric juices, or create a harsh environment through the use of the gastric juices, reduce the pH, make that condition very unfavorable for replicating bacteria. But then we want to make sure that we're neutralizing the acid as the contents leave the stomach. We also want to make sure we're emulsifying fats. We want to make sure that we dump the proper concentrations of enzymes through this duct from the pancreas in order to basically jumpstart that digestion process as we work through the duodenum into the ileum, where the absorption of the contents takes place. So the gallbladder is going to store bile that is produced by the liver, and the purpose of this bile is to reduce, or I should say neutralize, the stomach acid and increase the pH. So the pH of the stomach is a 2. The contents that are entering this part of the duodenum are coming from the stomach, and so upstream of this duct, the pH is a 2. Okay, the stomach contents leave the stomach. They're entering this duodenum, which is the opening of the small intestines. It's the part of the small intestines that connects to the stomach. It has a pH of 2, and as soon as the contents get here, where the bile is being injected or secreted through this papilla, it is neutralizing the acid, and so everything downstream of this papilla is actually going to be slightly basic at around a pH of 7.5, roughly 7.2. Okay, and that's important. Okay, we have to neutralize the acid as the contents of the stomach leave the small intestines for a couple reasons. One, we don't want to denature the enzymes that are being secreted at this particular spot by the pancreas. Okay, if the pH is too low for optimum, okay, the optimum conditions, the optimum pH of the enzymes, which we'll talk about in a minute, but if we don't produce an optimum condition for the enzymes that are secreted by the pancreas, then we're going to denature the enzymes as soon as they're secreted and render them completely useless, which is really bad news when we talk about digesting and absorbing food. So we want to neutralize the acid, we want to kind of return the pH to roughly 7.5, and we want to emulsify fats, both of which are roles of the bile, and that bile is stored in the gallbladder. Pancreas is going to secrete enzymes through that same duct, and those enzymes are going to kind of start digesting or hydrolyzing certain compounds, which we'll talk about in a minute, and that digestion will continue as the food contents work through the remaining duodenum before entering the back half of the small intestines, which are the ileum. So if we look at some of the hormones that are produced as kind of a secondary response to this digestion process, two hormones that we need to talk about right off the bat, and we'll talk about later in the IB syllabus, is insulin and glucagon. Now, both of these play a role in digestion, but are more related to actual glucose metabolism than true digestion. But some other enzymes that play a big role in this digestion process are lipase, amylase, and endopeptidase. All three of those are going to be secreted by the pancreas and are incredibly important as we digest food throughout the entirety of the duodenum. So, insulin and glucagon, like I said, both have or play a role in glucose metabolism. Obviously, insulin is a hormone that is released by the pancreas that causes blood sugar to be absorbed by the liver and stored as glycogen. Remember, glucose is a monosaccharide, glycogen is a polysaccharide, functional polysaccharide found in animals. It's the way that we store large amounts of glucose in our liver, and we store them in times of food availability for times of fasting. And so glucagon would be the exact opposite of insulin. Insulin causes blood sugar to be absorbed and stored in the liver. Glucagon is a hormone that is going to be released in times of fasting when blood glucose is low, and is going to result in glycogen being broken down in the liver and sugar being released in the form of glucose back into the blood, so that the cells constantly have a good quality supply of glucose. Really, really important. Both insulin and glucagon play into the homeostatic balance that must be present in order for humans and other organisms to remain alive. Lipase, amylase, and endopeptidase are all three pancreatic enzymes, which play a great role, or a big role, in digestion as a whole, and all three belong to the secretion that is called pancreatic juice. All three of those are going to be included into kind of a cocktail solution that is released by the pancreas through this papilla in order for the pancreas to get the enzymes into the duodenum so that digestion can happen. So, if we kind of look at the three enzymes, lipase, amylase, and endopeptidase, in detail, we know through earlier studies of enzymatic activity, if you haven't seen my earlier videos on enzymes, make sure you check those out. Lipase is an enzyme that breaks down lipids. Amylase, just like salivary amylase, is an enzyme that is going to break down starch. And endopeptidase are enzymes that break down polypeptides. Now, I'm going to focus on trypsin. Trypsin is an example of one form of endopeptidase. It is not the only endopeptidase, okay? So, I'm going to focus on lipase, amylase, and trypsin. We know from earlier enzymatic studies that enzymes have substrates. Those substrates are going to bond to an active site, which is a specially created site on the enzyme that is conducive to a substrate binding. The substrates for lipase would be fats and oils, basically triglycerides, and the lipase is going to break those triglycerides down via a hydrolysis reaction into the monomers that make up those triglycerides, which is the glycerol and the 3 fatty acid chains, right? So the hydrolysis of lipids will release the monomers, glycerol and fatty acid. It takes a lipase to do that. Amylase, specifically pancreatic amylase, is going to break down starch, but it's not going to break down starch completely to glucose. Okay? Amylase breaks down, or via a hydrolysis reaction, will break down starch into maltose. Maltose is a disaccharide that is composed of 2 glucose monomers bound together. It takes a different enzyme to break down maltose into individual glucose. The enzyme that does that is called maltase. Okay? Maltase is an enzyme that breaks down maltose. So when we talk about carbohydrate digestion, amylase breaks down complex starch polysaccharides into individual maltose disaccharides. Then maltose is broken down into individual glucose monomers using an enzyme called maltase, kind of as a secondary process on that carbohydrate digestion as a whole. Trypsin is an example of an endopeptidase, which is going to break down large polypeptides. Okay? These endopeptidase molecules or enzymes are going to break down really long polypeptides. Again, remember, polypeptide is another name for protein. And it's going to break these long, really big proteins into smaller proteins that are more manageable for further accessory enzymes to break down. Further protein digesting enzymes then hydrolyze the smaller polypeptides into individual amino acids. Again, what is kind of the similarity between all 3 of these enzymes? We are employing them to break down a specific substrate into specific products, but these specific products that are ultimately made end up always being monomers, right? A glycerol is a monomer. Fatty acids are monomers. Glucose, monomer. Amino acids, monomers. What are we going to do with all of these individual monomers? Well, monomers, as you hopefully recall from, again, previous videos, monomers are the building blocks of complex polymers. And so if we break down through digestion all of the complex polymers that we consume through ingestion into these individual building blocks, we can then absorb them easily through our cell membranes, okay, because smaller molecules can diffuse easily or more easily and also more efficiently through a cell membrane than larger molecules can. Okay, so we break these large complex carbohydrates and large complex proteins down into individual monomers, then put these individual monomers into our bloodstream, transport them to individual tissue cells, allow the tissue cells to absorb them, and then assimilate them into their own specific cellular needs, and basically use those building blocks to build up larger compounds that they need in order to maintain their life. The human digestive system, like we said, employs a variety of enzymes. Okay, this is a different diagram than what you saw in the first video, but I want to use it to kind of outline where you see specific digestive enzymes employed and what the relative pH or optimal pH is of those enzymes in that particular location. So, just like the digestive system in the first video, we're going to start with the mouth. Remember, salivary amylase, or saliva, that is produced by salivary glands is going to contain an enzyme called salivary amylase. Amylase, just like pancreatic amylase, is going to break down starch. It's going to hydrolyze starch into a product called maltose. Maltose will then further be broken down or further hydrolyzed into glucose molecules using an enzyme called maltase. The optimal pH of your mouth and the saliva is 7 to 7.8. About 7.5 is right in the middle of that optimal range. Okay? That optimum pH is ultimately going to maintain the optimal efficiency or optimal enzymatic activity of the salivary amylase. So you actually start the digestion of starch in the mouth. A quick experiment that you can do at home is you can take a saltine cracker and you can, without swallowing it, chew it up for a while. Okay, really mix in saliva, allow the saliva to kind of penetrate and to really kind of make the cracker soggy. You will notice, if you are making quality observations as time goes on, that the sweetness of that cracker actually increases over time. And the reason you're perceiving the sweetness to increase is because you're actually giving your salivary amylase time to work in order to break down the starch, which is contained within the cracker, into individual maltose molecules, disaccharides, which you, using your taste buds, actually start to perceive as sweet. Okay, maltose is a sugar that is actually sweeter than starch. And so over time, as those enzymes begin to break down the starch into maltose, you will actually perceive the sweetness as that enzymatic reaction takes place. Again, that is in your mouth, optimal pH 7 to 7.5. If we progress down the esophagus, we immediately enter the stomach. This is where proteases, okay, are finally are employed. Proteases are enzymes that break down protein. An example of a protease would be pepsin. Pepsin breaks down polypeptides. Polypeptides being proteins. Okay? How does it do that? It breaks these polypeptides into individual monomers called amino acids, and the optimum pH of this particular protein is 2. That is incredibly acidic and way more acidic than the optimal pH of your saliva, which is 7.5. When you swallow food and the food contents within that bolus make its way down the esophagus and enter the stomach, the salivary amylase, which is the enzyme that is contained within the saliva, is immediately denatured because the optimal pH of 7.5 isn't maintained. So the stomach essentially stops all starch breakdown abruptly at that point. Okay? Then it gives the stomach acid a chance to kill bacteria, if any exist, on or in the food, and it also works in the pepsin, or the proteases, which begin to break down the proteins. So already we have actually started to break down the starch in the mouth. We are now breaking down the proteins in the stomach, and then we're going to secrete the food products out of the stomach into the duodenum, where the next round of enzymes will be employed, and those enzymes come directly from the pancreas. The pancreatic lipase is an example of an enzyme that is produced by the pancreas and will be secreted by that particular duct called a papilla. There are a variety of enzymes that are going to be secreted in the same way. Obviously, lipase is one example. There is a pancreatic amylase also in this example. And then there are other specific endopeptidase molecules that are going to be secreted as part of that pancreatic juice as well. In all cases, the optimal pH of these pancreatic enzymes are 7.2 to about 7.5. Okay, that's really resemblant of what the mouth was. Okay, and so the pancreas is going to make those enzymes. It's going to secrete those enzymes downstream of the stomach, which is really important because if these enzymes were secreted into the stomach obviously isn't in the 7.3 optimum pH range, right? And so if these enzymes were secreted into the stomach, they would immediately be denatured, they would be immediately shut down, they wouldn't work, which, you know, in terms of human digestion, that would be terrible. Obviously, this is the gallbladder. The gallbladder would be connected via a duct to the same papilla, and so we would neutralize the acidity as it leaves the stomach so that we can return the pH back to the 7.3 range so that our pancreatic enzymes are back into their optimal range. Okay, so these are just three different examples, both, or all three, the mouth, the stomach, and the pancreas, that all have three different optimal temperatures. They're all going to secrete different amounts of enzymes. They're going to secrete different types of enzymes, all of which have different roles. Okay. Chemical digestion of starch begins in the mouth through the saliva, right? That's the salivary amylase. With the addition of saliva and the amylase contained within the saliva to the food, starch is hydrolyzed, or is beginning to be hydrolyzed into maltose. The enzymatic activity of salivary amylase ceases in the highly acidic environment of the stomach. Okay, we talked about that. Leaving the starch remains largely undigested through the stomach until the contents kind of meet the next round of amylase, which is when the pancreatic amylase is added post-stomach, okay, which is when it is released into the small intestine. Within the small intestine, there is another enzyme that completes the digestion of maltose. It's called maltase. We've talked about it, and it is secreted again within the small intestines, and it produces the two molecules of glucose from the single maltose disaccharide. That's where we're going to end it in terms of enzymatic activity. If you have any questions, make sure you bring those to class or see future or previous videos. See you later. Bye.